Hi, I'm Nicola Bordin, and I'm a research fellow at University College London, where I work in the CATH resource. So, what is CATH? CATH is a protein structure classification resource that over the past 25 years processed, classified, and curated over 500,000 domains into homologous superfamilies. So we take then this data and we expand the structural superfamilies with sequence information. And we do so by taking the structures and encode the motifs into sequence profiles and HMMs, and then we scan the protein sequence space using these HMMs, and we're able to expand this 500,000 domain structures to over 151 million domain sequences. So 1 year ago, Google DeepMind revolutionized protein structure prediction when they entered the CASP competition, and they made their code available and a collection of models available through the AlphaFold protein structure database in collaboration with EMBL EBI. And the initial release of 365,000 protein chain models was for 21 model organisms, with subsequent releases for Swissprot and organisms causing neglected tropical diseases. There is also a rumor going around that they're going to release models for the whole of UniRef 90, and they're coming in the future at some point. So as CATH, we are very interested in classifying this treasure trove of data. But if we look at our current protocol for assigning domains, we currently check weekly if new protein chains are deposited in the PDB, and then we retrieve them and we chop them by getting some domain boundary assignments, and then we assign these domains to a CATH superfamily, and all of the hits that get then manual curation by some expert curators in our teams. In terms of CATH domains, though, the AlphaFold2 dataset, the first release, is almost equivalent to 15 years worth of structures in CATH, so the current approach is unsuitable for this kind of dataset. So, in order to tackle this data, we came up with a new protocol called CATH-Assign, where we take chopping and assignment from hidden Markov models and machine learning-based methods, and then we validate these assignments using a cascading approach, using structural aligners to deal with these varying degrees of homology, and then we combine, in this case, established methods such as SAP, which is our internal structural aligner, and then new and ultrafast aligners such as Foldseek. So this has been quite the ride, because we were able to go from algorithm design, benchmarking, data processing, assignments, and curation for around 15 years worth of CATH structures in around 8 months, and also puts it in a very good place to cope with further releases of AlphaFold database now that the infrastructure is in place. So I'm going to go over the rest of the protocol later on during my talk, but the first step, it's by assigning domains to initial CATH mappings, since as part of our the creation of our sister resource, Gene3D, we have hidden Markov model hits for CATH domains and PFAM domains. And then we feed this kind of information to CATH-Resolve-Hits, which is a tool that we created that finds the optimal set of high-scoring and non-overlapping domains. And then we match them to CATH HMMs and PFAM HMMs, or another kind of domain that doesn't have either a CATH domain or a PFAM assignment, and we call them new FAMs. So for these last two type of domains, so for PFAM domains and new FAM domains, we don't have a CATH assignment at the moment. And they can't be detected by hidden Markov models. So they are probably very remote homologs. So in order to deal with this kind of data, we teamed up with the ROSLab team at the University of Munich, where we created a new predictor called CATH-E, which is based on embeddings from protein language models. And this kind of outperforms HMMs for most of these tasks. And especially for the largest superfamilies, we're able to get predictions correctly almost 90% of the times. And especially when compared to other state-of-the-art models, methods, such as other predictors or BLAST or even hidden Markov models, we're almost always able to outperform these methods. So once we have these assignments, then we use this cascading approach that for CATH hidden Markov model matches, we we scan against the CATH-S95 representative that brought this sequence in. And for other domains like the PFAM and NewFAM assignments, if they have a CATH-E match, then we scan against the superfamily that was predicted by CATH-E and by using Foldseek, and then we validate again using SAP. And then if by chance we are not able to validate this particular assignment against the superfamily, then we use this cascading approach where we scan against the fold, all the superfamilies within that fold, and all the folds within that architecture, and so on, until we reach to the point that we scan against the whole of CATH in the most complicated cases. We also notice that for some domains, in this case, we actually need to scan against CATH plus all the domains that we were able to bring in the previous steps. So while this is the kind of protocol that we used, we kind of had to introduce some kind of filtering criteria because we're still talking around 800,000 domains in this case. So we are interested in bringing in PDB-like domain and domains that are similar to things that are already in CATH from AlphaFold into our database. So we need to set up some pre-filtering criteria, not only to trim down computing time, but also to discard structures that don't look like globular domains. So we set up various criteria for filtering. First of all, we look at model quality. So we look at the average PLDDT for the whole domain. If it's less than 70, we discard them. And then we tend to discard structures that are very tiny. So if they have less than 3 secondary structure elements, they get also discarded. And then we look at residues that don't have a secondary structure assignment. If more than 65% of the structure doesn't have a secondary structure assignment with the SSP, then we discard them too. And then we look at the presence of long stretches of unordered or low quality regions, as well as packing in the domain core. So if we look at the composition of the discarded domains, we can see that almost half of them, they get discarded because they are of low quality, with an additional 23% that they are very tiny domains with less than 3 secondary structure elements. And we can see that the distribution is more or less similar for all the other 21 model organisms, with the global distribution on the left for the whole AlphaFold dataset. So with that taken into account, if we combine it with the good quality models, we're still processing 52% of all the domains available in the dataset, which is around 370,000 domains. And this is it's a number that if we have to scan against the whole of CATH and against all the superfamilies, especially if we expand them with AlphaFold, it's unfeasible. So SAP is very precise, but it's also very slow in this case. So we teamed up and with the Foldseek team with Foldseek, created by the Steineger group at the University of Korea. And they created this incredible tool that, while being 20,000 times faster than kind of state-of-the-art aligners like TM-Align or SAP or DALI, this is, with further and subsequent releases of the code, they are now comparable in precision to TM-Align. So this helped us massively in doing this kind of assignments. So in order to test this particular tool, we had to set up a sensible benchmark for this. So we took a dataset of 3,186 domains that we call the CATH-Scope Golden Dataset, where we have equivalent boundaries shared between and the same superfamily annotation, both in CATH and in SCOP. And then by knowing what's their relationship between the superfamilies, if they're homologous or non-homologous for these kind of domains, we were able to set up some thresholds to detect homology in using these tools. Since we noticed also that these behaviors, these tools, they behave differently depending on the CATH class. If they are mainly alpha, mainly beta, or mixed, we had to come up with different thresholds for different classes. So once we set up these tools, we perform all this cascading assignment, and we were able to expand the CATH dataset by almost 360,000 domains. And we can see that the vast majority of these hits, they enter through are very remote, are very close homologs, and they come from CATH-HMM hits. But then we can still see that there is an additional 8% that are outside of reach of hidden Markov models, and we can only assign using this new predictor that we created, CATH-E. And then there is a further 10% where we didn't have a CATH-E prediction, but we were able to validate it using Foldseek or SAP by scanning against the whole of CATH expanded by AlphaFold models. And then there is also another 8% of domains that are remaining and we couldn't assign to CATH, but I will go back to them later on during my talk. One of the advantages of this is that we notice that the organism expansion by AlphaFold2, it's incredible, especially for certain organisms where glycine max and rice, we were able to expand almost 4,000 folds their structural coverage. And this also helped us in the long term also for other kind of analysis that we do and other assignments that we have in CATH, such as the functional families. So functional families are, although the structure is conserved, there is enough sequence divergency in CATH superfamilies that we have different functions in these structural superfamilies, or as we call them, we have functional families within superfamilies. So FunFams are basically functionally coherent subsets of sequences within a superfamily that we extract and then characterize, and then we're able to segregate these kind of different functions based on residues, differently conserved residues in the alignment. So functional families are great for doing function prediction and lots of other applications, but one of their downsides is that they only have 8% of these FunFams, they have a structural representative. So using AlphaFold models, we were able to increase the structural coverage all the way up to 30%. So on the next slide, I'm going to show one of the examples that we were able to elucidate using this, and it's part of the HUBS superfamily, where by using a very good AlphaFold2 models with a PLDDT over 90, we were able to inherit from the closest neighboring functional family structurally determining position in this case, and specificity determining positions. And by that, we were able to inherit this kind of annotations and mark all these differentially conserved residues in this case. But now to go back to the remaining 8% of domains, these amount to around 35,000 domains that couldn't be assigned to a CATH superfamily. So in order to process them further, we did an all versus all comparison, and then we clustered them using T-cluster into 4,200 potentially new superfamilies. And then we clustered them, we characterized them and did some manual curation on it. We noticed that around 714 of these clusters, they consist of multi-domain proteins, and then 618 of them, they have at least one human proteins, and then we kind of focused our manual curation on those. And by looking at these 618, in the end, we ended up with 26 that were new folds, previously not encountered in CATH or PDB. So while the vast majority are kind of alpha bundles in this case, we also find some very peculiar fold arrangements where we have, on the bottom left, you can see a very peculiar alpha beta propeller. And on the right, there is what we call a hard domain, basically that it's involved in T cell activation in mitochondria. And on the PDB and for monomer structures, we can see that it can kind of adapt an open and close conformation depending on its current interactor. So these are the other 26 folds, as you can see here, and all of these domains are going to be made available, are available on Zenodo by Superfamily and by Organism, as well as the CATH FTP. Then individual models, they are going to be made available through the 3D Beacons network, and in the future they're going to be available through other venues. And currently we're processing Swissprot with neglected tropical diseases next. So, and with this, I would like to thank you for your attention, and I would like to thank especially Ian Insolito in our team for his help in setting up the chopping pipeline, Vamsi for his help with the CATH-E predictor, and Technical University of Munich team at the Rost Lab, Clemens for his help in setting up the packing, and Vishali for her incredible effort in manual curation, and Sudat Lam for his benchmarking and for PDB and CATH in this case. Thank you also to Stephanie and Martin for their help with benchmarking Foldseek. Well, thank you very much. I hope you enjoyed the talk.